Hello, welcome again to King's Bioproduction Academy. Today we're going to learn about the extension of the cell division. So that is our now second stage, which is meiosis. This meiosis occurs in the reproductive cells. Mitosis occurs in the somatic cells. So the reproductive cells, that is where we have maybe in males, in plants, we have in the anthers, and in the females, we have in the ovaries. That is the over here. Then in human beings, that is the XY chromosome, right? So let's discuss about the phases. Still, the phases will remain to be the same, but these will be undergoing under two stages. Stage 1 and stage 2. That is phase 1 and phase 2. So still it's the IPMAT process. That is interphase, prophase, metaphase, anaphase, telophase. But for this one now, we'll be having interphase 1 and the other one what? Having 2. But we don't, then we have what? Anaphase, the same way, interphase, prophase 1, 2, all of them will be running into cycles. So let's start with the interphase 1. What happens in the interphase 1? It's just similar with the other one. It's just a preparatory stage. What happens at this point? What are we preparing here? This duplication of the genetic material, that is 1. Number 2, still, we have the replication. That is the duplication, the replication of, or the synthesis. Duplication, sorry. The duplication or the replication of the genetic material. Then we have the buildup of energy, that is ATP storage form. Then that we will have what again? We will have synthesis of the new cell organelles. That is Golgi bodies, mitochondria, even the centrioles. From that point still, we still have the same things, like presence of nucleolus, 1. We have presence of the nuclear membrane, which will be in that stage. And the nuclear materials are just a thread-like called chromatin. From now, the interphase, we will go to the prophase. The prophase for this stage is very different from prophase. We are speaking about prophase 1. We had discussed interphase 1. Now we are in prophase 1. So in prophase 1, what happens? The same things that were in prophase of the other mitosis will still be here. And we had said this nuclear material, that's the chromosomes, were now shortening, thickening, and becoming more visible. Second, there was breakdown of the nuclear membrane, starting in breakdown, not complete breakdown of the nuclear membrane. That is the second. Then, the third, we have now other things here. The homologous chromosomes align side by side to form something called bivalence. And these one a process called synapses. And after these, there will be crossing over, leading to the genetic material being exchanged. Genetic exchange between the homologous chromosomes. So we are saying the first thing is what? Bivalent formation. That is homologous chromosomes lying side by side. A process called synapses. After that, what happens? There will be genetic exchange. Genetic what? Exchange due to crossing over. Because of the crossing over, we have genetic exchange, and we will be done with this prophase 1. And that is where we find difference in individuals. It's only in prophase 1 where it will determine the characteristics of an individual of an offspring. We go to metaphase. Metaphase will still be the same with that of mitosis. The only difference is metaphase 1, we are speaking about homologous chromosomes. So the homologous chromosomes will lie, not lying side by side, but will do what? Before that, on prophase still, there are some things that we have not said. it was about the formation, the spindle fibers. The spindle fibers were moving to the opposite poles, and there was formation now of the spindle fibers. At the metaphase, there is lengthening of those spindle fibers to attach the homologous chromosomes at the centromere. So we are saying the homologous chromosomes will align at the equator. They'll just form a line. They'll just align themselves at the equator. And when they align at the equator, what follows? What follows is the attachment of those homologous chromosomes at the centromere by the spindle fibers. And these spindle fibers, we are saying they have lengthened. From this point now, what happens? We go to the next stage, which is the anaphase. The anaphase, we speak about those homologous chromosomes. They move to the opposite poles. Homologous chromosomes separate, and move to the opposite poles. And this is due to the shortening of the spindle fibers. So our spindle fibers shortens, and because of the shortening of the spindle fibers, those homologous chromosomes are being separated and they move to the opposite poles. From that point, we go to the telophase. At the telophase till, in an animal cell, there is constriction of the cell wall. And after that, those homologous chromosomes now will be enclosed by nuclear membrane. There will be reformation of the nucleolus, and those cells separate to form two daughter cells. In plant cell, the same happens, but now the difference is there is no constriction, but there will be formation of a cell plate due to presence of a cell wall. The next now is about, from the telophase I, we have been discussing about interphase I, prophase I, metaphase I, metaphase I, anaphase I, and telophase I. Let's go to the second phase. Our second phase will be interphase II, of which what happens in the interphase II? That's just a resting phase. We don't have anything going there. It's the same things that we have discussed. So we'll just proceed directly to speak about just the yield of energy to be used in the successive phase 2. That is now Prophase II. What happens in all this stage that we are speaking about, either be it interphase II, Prophase II, Anaphase II, Metaphase II, Anaphase II, and Telophase II, they're just the same as that for meiosis. The only difference is we are running here in two circles, so that at the end we divide them so that we form 4 daughter cells. The mitosis process we undergone, there was division, and at the end we found 2 daughter cells. Here we are running in 2 circles so that we can get 4 daughter cells, but the process will just be the same. For the interphase, preparation, duplication of the genetic material, synthesis of the new cell organelles, build-up of energy, still the same. At Prophase II is still the same. What happens? From those 2 daughter cells which were being formed, what they will be undergoing, we form centrioles. That is one. Those are the nuclear membrane, the nuclear material, The nuclear material starts to do what? Those nuclear materials, we're saying they duplicate. After they are duplicated now, those centrioles will be formed. After they are being formed, what next is about the nucleolus disappearing, the nuclear membrane starts to break, and formation of those what? Formation of the spindle fibers. At the metaphase now, what happens? Metaphase II. In metaphase II, the same way to metaphase in mitosis, we say the chromosomes will align at the equator. When they align at the equator, those spindle fibers lengthen to attach those chromosomes at the centromere. That is our second. And we say there will be complete disappearance of the nuclear membrane. Then on anaphase, the same story. Anaphase II. What happens? Those sister chromatids from both of the cells, we are saying we are running into circles, they will separates and move to the opposite poles. And then this one, we're saying due to the shortening of the spindle fibers. At the telophase, still the same things undergoing. If it's an animal cell, there is constriction of the cell membrane. Second, we're saying those sister chromatids, they will be enclosed because of the reformation of the nuclear membrane. There will be reappearance of the nucleolus, and this cell separates now to form 4 daughter cells. The 2 cells will divide 2, 2, and they will form 4 daughter cells. If it's a plant cell, the same thing undergoes, but we form a cell plate. We form a cell plate. There is no constriction of the cells. Thank you. We'll continue learning more. And in case of any question, just you can ask on the comment section for more explanation and even understanding. Subscribe so that you can get more of my videos to learn. Thank you very much.